Germany, UK, Africa, Singapore, Bermuda, Italy, the Netherlands, and Norway as part of various academic and exchange programs. His contributions in biological oceanography as an academician and administrator are matchless. He was the coordinator of the national program in harmful algal blooms funded by the Ministry of Earth Sciences, Government of India, with the participation of several institutes, national institutes and universities in India. He has guided more than 40 PhD students in KUSAT. He has occupied several academic and administrative positions like Professor of the Department of Marine Microbiology and Biochemistry. And nearly he was the head of the department for 15 years. And he was the director School of Marine Sciences, KUSAT, for nearly 10 years. And he was the Dean Faculty of Marine Sciences, Dean Faculty of Environmental Studies, Member Syndicate KUSAT, Registrar in Charge, and Pro-Vice Chancellor in Charge KUSAT. He has also served as member and chairman in several academic bodies in national and international level. And after retirement from KUSAT in 2001, he has been serving as Emeritus Professor in the School of Marine Sciences KUSAT. And he has placed in eminent positions like Chairman, Nansen Environmental Research Center India, and Dean Faculty of Climate Variability and Aquatic Ecosystems, KUFOS, and Standing Council Chairman for Academics, KUFOS, Member Governing Council, and the Senate, KUFOS, and Subject Expert Kerala Coastal Zone Management Authority, Chairman, Research Advisory Committee of CMFR, ICR, etc. And his immense contributions to marine sciences include more than 200 research articles in reputed journals, 2 monographs in taxonomy, and several others. He was also a pioneering taxonomist of marine invertebrates and groundbreaking ecologist. In spite of being a scientist, he was a great thinker, philosopher, humanitarian, and a man of wisdom. And his sudden demise was a great loss to the marine science community, and he will be remembered by the scientific community of School of Marine Sciences, KUSAT, and oceanographers at national and international level forever. And it is very fortunate that during this occasion, we have with us Professor Ajmal Khan, who knows Professor N.R. Menon personally, with us to deliver the lecture today. And I should congratulate Dr. Bijoy for organizing this webinar in honor of Professor N.R. Menon when we are completing the 3rd year of his loss. And our association, Professor Ajmal Khan is Professor retired from the Annamalai University, and our association with the Annamalai University dates back to 1970s. I used to hear that from my husband, Dr. Bright Singh. He studied there only, and he used to tell Professor C.V. Kurian, Professor Balakrishnan Nair, they used to go there, they used to come to Annamalai University as examiner, and they used to deliver lectures there, and they used to have, in the night, they used to have dinner together. So it was a wonderful time. He used to tell me. I know Professor Ajmal Khan sir also was there. And our association with Professor Ajmal Khan sir actually started with in the early 2000s when we started the multi-institutional Bendik Productivity Program funded by the Ministry of Earth Sciences, where Professor R. Damodaran, the world-renowned Bendik scientist, where he was the coordinator of the program. And doyens like Professor Ajmal Khan sir, Professor Ravi Raman, Dr. Sarala Madam, and Dr. Ansari were all collaborators, and we used to meet together in various conferences and project assessment meetings. And Professor Ajmal Khan is a frequent visitor to our department as resource person in seminars, symposia, as well as the various training programs, especially with respect to our this primer. And he's the patience and the perseverance is remarkable, Professor Ajmal Khan. And besides being a doyen in marine biology, his expertise in marine benthos and impact assessment studies are well known. And he is a very busy consultant to various institutions for impact assessment studies in India as well as abroad. And even after retirement, he is very active in science, and his passion for science and inquisitiveness to learn new things, especially the primer and all statistical analysis, those qualities have attracted me a lot. And we are very, very fortunate that we have with us Professor Ajmal sir to deliver this lecture. And one more thing I should say, we have to learn a lot of things from Professor Ajmal sir. Besides being a doyen or a highly resourceful person, he is very humble, simple, and respect to elders and colleagues. So we have learned a lot of things from you, sir. Such a humble person. So sorry, agreeing with that. Okay, we are really fortunate that we have with you, sir. And
Thank you.
on behalf of all of us, I invite you, sir, to deliver this lecture on monitoring of marine environment. Over to you, sir.
Thank you, madam. Thank you. Thank you, madam. Thank you, madam.
Thank you. Thank you,
Thank you,
madam. Thank you so much. Yeah, sir, please.
So pleasant. Good morning, our respected leader and the distinguished dean of CUSAT, Professor Rosama Madam, very dynamic head of the department, Professor Dr. Vijay Nandan, our teacher, Professor Sarama Madam, Dr. Surjit, Dr. Vadmakumar, all the learned faculty members of CUSAT, in particular Department of Marine Biology, Microbiology, and Biochemistry, and all my young friends. I am extremely thankful to Professor Rosama Madam for introducing me and for all the nice words. I still continue to be the student of all of them, Dr. Rosama Madam, Dr. Sarama Madam, Professor Damodaran, Professor Vijay, Professor Hatha, all. I used to learn many things from them. Whenever I meet them, I hear them, I discuss with them. So I am extremely happy. I am happy that this lecture, it has been arranged in memory of a legendary scientist, a doyen scientist, Professor Menon. Professor Menon was a very warm person, very kind person. As Madam pointed out, he was having a very rich scholarship. He used to be nice when we converse on personal sides. But when we used to discuss science, he never used to mince words. He will not mince words in calling spade a spade. So that was very good for youngsters like me. That time, that was very good. On personal side, how we have to talk, and when we are discussing science, how we have to discuss, all those things we learn from him. I feel happy that this lecture is coinciding with the 3rd anniversary of his passing away. With the due respects, I pay my homage to him and start my presentation. He served the cause of the environment. he was carrying out quite a lot of very good studies in pollution and monitoring. So that way this talk, it is also on monitoring of the marine environment. So very good coincidence, but for this coincidence, Professor Vijay is wholly responsible. I thank him warmly. So I am starting my presentation now. So are you able to see the slides?
Not, sir.
No, sir. No, not it. Please,
So shall I get some help from there?
yes, sir.
Ah,
Yes, yes. Present now, you can click on the, open the slides, sir, and put it on the slideshow mode, and then press on the present now. There is a cap.
At the bottom right, you can
So
see
present is
now.
here.
Present.
Ah,
Yes. First, open your
Present is
slides,
there.
sir. Open your PowerPoint.
Yes, I am
sir.
opening my slides.
Open in, put it in the slideshow, sir. Slideshow.
Oh, right,
Then,
right. That is not visible.
then
The
you
slideshow is not visible.
no, sir. Put it on present now. You have to press, sir. There is
present now.
Present. Yes, I have. Your
And then
entire
on
screen.
the top, you
Your
can
entire
see
screen.
where-
I am. Am I right?
no, sir.
No,
There is
no, not that.
at the bottom on the right side, there is a present now.
Yes,
indication.
yes, sir. Yes, sir.
Present. Press on that, sir. Then it will
Yes,
ask.
yes, I am doing it.
When you
So
ask for
present
present,
now, I am.
Do you-
So
you
now
see any
present
window?
is there,
then
and
Then
your entire screen is there on the top. Below that, a window is there.
then you
Then
ask.
below that,
then-
a tab is there.
yes, sir.
one more click on that top
Sir, it
window.
is a window.
Oh, right. Top.
Window, one more click in that top window.
Yes, madam.
Then share at the bottom.
Yes, share. Yes.
Yeah. Then open your PowerPoint.
Yes, madam.
PowerPoint. Yeah, it's coming.
Coming. Fine.
Okay.
It is coming. It will take just that
Yeah,
time
1 or 2
lag
seconds.
will be there. That time lag.
Oh, right.
It's coming.
So, shall I start now?
Wait, sir.
It is coming.
Okay.
Not that you are presenting. Your PowerPoint, have you taken, sir? Your PowerPoint?
Yes, madam. I have started sharing.
In your laptop, it is the desktop. You can see that PowerPoint slide.
Yes, madam. I am able to see, but on the side, I am not able to see me.
Okay, but in our screen, it hasn't come. Sir, no, sir.
It does not come, teacher. It does not come. It has
Right.
stopped sharing. It has stopped sharing.
Oh, it is.
Please do it once more, sir. It will come.
Okay, sir. Okay, sir. Yes.
Please do it once more, sir.
So you are presenting. I am clicking this one.
Yeah.
So is it visible, sir?
No.
No.
It has not started
Is
sharing.
there anybody else there? You can take the help of them if there is anybody else.
Or else,
Does it on, sir?
Yes, madam.
And I think you can do it once again. Now stop presenting. You can do stop presenting.
Stop sharing.
Yeah. Stop.
Stop sharing.
Then
And
now once
again, I am.
Yeah. Once again, please. Stop.
Again, madam.
Again, sir.
Again?
Sir, PowerPoint slide is open, no? In the desktop, open.
It is open, madam.
Okay, slideshow mode. Okay, then in the Google Meet window, present now.
press on.
Yes, present now. Yes, madam.
Now at the top we can see a window, no?
Yes, madam.
There, I think something will come, share or something, that window.
Yes, madam. Show everyone.
Yes.
Chat with everyone. So shall I press this one? Chat with everyone.
No,
Okay,
share.
sir. Yes, sir. Now at the bottom we can see share. Are you seeing share?
Shall I take some help, madam?
Yeah, yes, sir.
Okay, thank you. Yes, thank you. Sorry for that.
No problem.
உட்கார்ந்து ஸ்லைட் ஷோ பண்ணா உட்காரலாம் கஷ்டமா இருக்குமா இதை வச்சுக்கலாம் அவங்க பேசுறது ஸ்கிரீன் ஷேர் ஆமா ஸ்கிரீன் ஷேர் ஸ்கிரீன் போடணும் பேசுறாங்களா Are you able to see, sir?
Yes.
Yes, fine.
Yeah. Shall I start then?
Yes, please.
Okay, sir.
Slideshow mode.
Slideshow, can you put it in the slideshow?
Slideshow mode.
5-star. 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 The slideshow mode.
Presentation mode.
Yes,
And
ma'am. 1 minute, madam.
Icons on the side will go.
Slideshow. Slideshow என்ன தெரியல அதுல வருமா from beginning.
Yes, fine. Excellent. Excellent, sir.
Okay, sir.
Excellent.
Thank you. Sorry for the delay.
It's okay. Please take your time, sir.
Yes, sir.
Thank you.
So the marine environment, the world ocean covers about 70.73% of the Earth's surface. So appropriately, it has been described as the immeasurable immensity and also as invulnerable immensity. Thanks to the technological advancement, we could measure the length, breadth, and also the depth of the world ocean. So that way, it is no longer immeasurable. Due to the anthropogenic activities, I mean due to the human activities, the ocean is showing many changes. So that way, it is no longer invulnerable. But in view of the manifold uses of the marine environment, we have to monitor the ocean environment. So what is environmental monitoring? Very simply, we can put it as recurrent and systematical investigations or observations that show the status of the environment. So if anybody is asking for what purpose we have to monitor the marine environment, we can explain with these 3 points. So what for marine environmental monitoring? To describe the status of the marine environment. So through monitoring, we will be able to find out the status. We will also be able to follow the changes that are taking place in the marine environment. And also we will be able to follow the effects of the measures applied. What I mean is, the environment we have been concentrating, we are able to see adverse changes. To rectify that one, we are undertaking some remedial measures. So what are all the result of those remedial measures we have applied? So to follow up the effects of remedial measures applied also, we can undertake the marine environmental monitoring. So where lies the need for monitoring? So the marine environment has the vast potential. So with this vast potential, it plays a significant role in the economy of the nations, not only the coastal nations and also the inland nations. It is having so much. Moreover, it is the moderator of the world climate. In the absence of ocean, what will happen during the winter season? It will be much more chiller in the absence of the ocean. And also during the summer, it will be much more hotter. So that way, the world oceans act as the moderator of the world climate. And the world trade, it is impossible without the sea routes. And all of us are in the know of things. All the bulk materials, these are being moved through the sea routes only. Very urgent items like COVID vaccine and other things, we can airlift. Other important items we can airlift. And bulk items, oil, food grains and all, cars, we may not be able to move. It will be too expensive. So that way, the world trade, it will be impossible without the sea routes. Through the clear waters, through the clean waters, if you are carrying out the trade, it will be well and good. In Cochin backwaters, we are able to see the movement of quite a large number of vessels. Comparatively, the water quality is clear. Same way, we may not be able to have any traffic in Kuvam waters of Chennai. It will be a nauseating experience if we attempt such an activity. So that way, the world trade, it is impossible without the sea routes. So we have to undertake monitoring. We have to keep the waters quite clean. So marine environment is playing a predominant role in the well-being of people through its provisional and supporting services. What I mean by provisional services, here we see the importance of food, medicine, seafood. It is having quality. Medicine. Modern day man is afflicted with quite a large number of diseases. Around 70% of the modern medicine, it is coming from biodiversity, and in that large proportion from the marine realm. And many more we are in the process of getting. Clean air and other things are coming under provisional services. Then we have the supporting services. Oil, from where we are tapping the oil. Gas, we tap quite a large quantity of gas from the world oceans. Tourism also. Many island nations are attractive destinations for the tourists. So that way, millions of tons of oil are produced from the shallow waters of many seas and also oceans. So every day, how much we tap? We tap as much as 8.2 crore barrels. So one barrel, it is equivalent to 160 liters. So much we produce thanks to the marine environment. It also happens to be the food we are getting from the sea. It happens to be of top quality. It has top-notch protein of high biological value, which is very good for nourishment. It is also having high concentration of all the essential amino acids. And these essential amino acids, they produce the required protein and also other substances. They provide energy for exercise and other things. They form the active sites of enzymes. They strengthen the immune system, and they are also serving as antioxidants. So quality is there, very good quality is there. That's why the Food and Agricultural Organization, it has described the food from the ocean as the nature's superfood. Please have a look at the equation given below. 100 gram of fish, it is equivalent to 155 gram of egg. So fish is superior than egg. Further, it is equivalent to 165 gram of wheat. Further to 285 gram of rice and 500 gram of milk. So that way, the fish is superior than egg, superior than wheat, superior than rice, superior than milk. But rice and wheat, they happen to be our staple food. Whether we'll be able to leave our staple food elsewhere, they have been doing. They buy a large fish, they divide it into two. For one meal, they have one half, and for the other meal, they finish the other half. So food habits can be changed very slowly. Of course, overnight, it will not change. But the quality of the seafood is good. It is good for curing so many diseases. So that way, the purity has to be preserved, and that also requires monitoring. So the marine environment is providing us with oxygen. The oxygen you are inhaling in Kochi, the oxygen we inhale in Parangipete, courtesy the marine environment. So what is happening? The phytoplankton, which drift in the ocean waters, they do photosynthesis in the presence of sunlight. And they make use of the carbon dioxide available in the atmosphere. The end product of photosynthesis is oxygen. So it is this oxygen you and me are inhaling. It is providing us with water also. Again, the phytoplankton, what is happening is, they produce a sorry, a secondary metabolite called as DMS, dimethyl sulfide. It is for meeting their physiological requirements. The excess produced is rising to the atmosphere. It is acting as the cloud condensing nuclei. It gathers moisture and forms the rain-bearing cloud. Coupled with wind action, it is raining in various places. Recently, we had a very good rain thanks to the phytoplankton and also the activity of the wind. So through the hydrological cycle, we are getting the water, and phytoplankton is playing an important role. Thanks to the biogeochemical cycle in which the vast republic of microorganisms are playing a very active role, they decompose organic matter, they recycle the nutrients. That way, we have a very productive marine environment. We also have a serene and clean environment. So all due to organisms living in the marine environment. Sorry. So this is the hydrological cycle, and this is the biogeochemical cycle. This cycle, it is incomplete without the decomposers, that is the vast republic of microorganisms, both on land and marine environment. They are very much present and they play a very good role. And in the marine environment, we have immense quantities of various salts and chemical elements. Even gold is there. Generally, American scientists used to do many funny things. They used to do very good work, no doubt. They used to do funny things also in between. But it happened to be the turn of a Russian scientist. He calculated how much gold is available in the world oceans. Then he did a calculation. Suppose he is dividing to the world population. He tried to see how much each and everyone will be getting. He arrived at a figure of 4 kg. So I will be getting 4 kg and all our participants will be getting 4 kg each. So that will be at current price, it will be a very fabulous. But how much we have to spend? We have to spend much more. That's why we are not attempting. What I am coming to say is, even gold is there in the seawater. And the era of commercial exploitation of the ocean bed resources, it is fast approaching. Many important metals are available in the seawater. I will cite only 2 examples. Take for instance the nickel. Nickel we require for making the batteries, for making the cell phones. In buildings, it is being made use of. In transport, it is being made use of. And also, in many other things, it is being made use of. So, how much nickel is there on land? For how many years we will be able to exploit? For another 100 years at the current rate of exploitation, we will be able to do it. But coming to the marine realm, the quantity available is 15 billion tons. So for the coming 1,50,000 years, we don't have to worry at all. And with respect to manganese, manganese is an alloy, very good alloy of steel and other things. Please look how much quantity is there. 358 billion tons. So the manganese on land, it will be lasting only 100 years. But if we start exploiting the marine resource, for the coming 4 lakh years, no worry at all. So much potential is there. In all these things, what is important is the quality. So that quality should not get impaired. So for that we have to do the monitoring. Tourism. Many island countries, they are earning fabulously through seaside tourism. In India, Kerala is the #1 state. People from different countries, they throng the hill resorts and also the seaside tourist spots. Only when the water is clean, the beaches are clean, people will be coming in large numbers. A country which is present on the southern side of our country, Maldives, it earns fabulously through seaside tourism. So they have to maintain the quality of the water for bathing and other purposes. So that also requires monitoring. You might have heard about the movement, establishment of the Blue Flag Beach. It is concentrating on the quality of the bathing water in the selected beaches and also marinas. So what is the difference between the beach and the marina? Cherai beach only. Marinas means yachts will be there. Making use of the yachts, we will be able to go to the various places. We can go for diving and all. So this movement, it started in the later part of the previous century. It started in France. They certify the beaches. They give the credential as Blue Flag Beach. There are many tourists who look for this emblem, who look for this recognition. And such seaside resorts are having a very good attraction to the world tourists. So we have beaches all along. If you are able to establish Blue Flag Beach all along our coast, then through tourism also, our country will be able to earn quite fabulously. So here what is important is to get this certification, the monitoring is important. Even microbial monitoring is important. The water, the bathing water should not be infectious to the bathers and all. So this requirement is also there. So this is the marina you are able to see. So now we know the difference between a beach and the marina. So many yachts are there waiting for the tourists. So as I mentioned, near Kochi we have the Cherai beach. Very good beach I have visited once with the family. There was quite a lot of rush. Many people were coming enthusiastically. If this beach is having that certification, it will add to its quality. There is another beach which is having this certification. That is present near Kozhikode. The place is called as Kapat. So high time, the Cherai beach is also getting. But what is very important for getting this certification is marine monitoring. We have to monitor the water quality and other things. So we are able to see the increasing incidence of storms and also cyclones. How much damage the storms will be able to wreak, we have been seeing from time to time. If you are in a position to predict the occurrence in advance, we will be able to alert people. That way we will be able to avoid large-scale destruction of men and materials. So this also requires monitoring. Only through monitoring we will be able to give the forecast. Then there is periodical occurrence of harmful blooms and all. Blooms are happening due to various organisms. Trichodesmium is causing the harmful algal bloom, and other organisms are also causing the harmful algal blooms. What is happening is they deplete the oxygen. And immediately there is mass mortality of the fishes, leading to economic hardship of fishermen. So we have to avoid. Suppose we are able to predict the occurrence of the harmful algal bloom, and such catastrophic events can be avoided. So this is another instance of mass mortality of the fishes. And now I like to tell one incident which happened in the Persian Gulf area. In the Persian Gulf area, the traffic is quite heavy. You know the reasons for that one. And we have been importing oil mainly from the Gulf countries. And in turn, Gulf countries, except oil, they are importing everything. So the traffic, ship traffic is quite heavy. In one port, large number of vessels were anchored. One fine morning, they could not start their vessels. They could not run the blades. They could not operate their water pipes because the whole thing was clogged by the blooming of a pteropod. We call it Chrysis acicula. It is popularly also called as sea slug. All of a sudden, there was a blooming. Suppose the ports are monitored regularly, and we could have avoided such an eventuality. So this is also explaining. And who identified this organism there? This event happened in the Gulf. And who identified this organism as pteropod Chrysis acicula? A scientist from Kerala identified this one. So that is the credit of your state. So coming to the, again, need for monitoring. So you are all in the know of things. We do know that rapid changes are taking place in the environment. No doubt the changes are taking place since billions of years. The age of the earth is 4.5 billion years. The age of the ocean is 3 billion years. Since its birth, one or other changes are taking place. However, if you look at the present, the scale, the magnitude, and the speed at which the changes are taking place, in particular after the Industrial Revolution, especially over the last 60 years, it is new and it is also worrying. And only through monitoring we will be able to rectify the situation. So the human activity is so much that it is leading to increased risks, and in many instances the critical thresholds have been crossed, resulting in drastic changes. We are able to see these drastic changes throughout the world. So all these things necessitate marine monitoring. So if you look at what are all the drivers of environmental changes, population is increasing. Even there is an explosion in global human population. We have to check the rise in population. In correspondence with the explosion in global human population, there is the economic activity. And the technology is going on improving. We are improving the technology, and that is also having a telling effect. Then the social behavior. Man is slowly changing his behavior. He is moving towards sophistication. His preferences are changing. And in accordance with that one, we have the governments. We have the populist governments. They are not worried about the environment and all. And they want to please the people, and they want to keep up the World Bank. So that way, politics is also playing dirty, and there is lack of governance. So all are the drivers of the environmental changes. And these are all the environmental determinants. Pollution, it is the number one problem, particularly the nuclear pollution. To augment power generation, We have been setting up nuclear power plants all along our coastline. That is heading to the O. They are starting as one unit. They are adding another unit, then third, fourth, like that they are going. And even though people are resisting, still to meet the power requirements, government is having no other alternative other than setting up the nuclear power plants. Then we use quite a large quantity of electronics, and many things are ending up in the garbage. So how to dispose of? And plastic, it is also another menace. Microplastic, one of my friends mentioned that it has reached the world oceans to the depth of 10,000 feet. So it is going on invading the entire marine environment. So what will be the future, we do not know unless we are checking it. Global warming, due to the lifestyle, the globe is getting warmed. There is the ocean acidification. We are seized of this problem also. There is depletion in the ocean layer, and we try to construct so many things, and the habitats are degraded. When habitats are degraded, there is loss of biodiversity also. So all these are determinants. So to maintain the pristine purity of the marine environment, at least to stop further slide, we have to undertake the marine monitoring. So in such a grave situation, in such a background, environmental monitoring plays a crucial role in checking and preventing the further slide. Coming to the methods which are being used in environmental monitoring, most of us are biologists. There are many very good biological methods making use of which we will be able to monitor the environment. We have been making use of physical-chemical parameters also. So through those means also we will be able to monitor the marine environment. So first we will see the biological methods, then we will move on to the other methods. So coming to the biological monitoring, we make use of the entire trophic levels as indicators of environmental change. We make use of phytoplankton, in particular, the concentration of chlorophyll or the biomass as indicators of eutrophication. Eutrophication, we have to monitor, we have to find out, we have to predict, then only we will be able to avoid mass mortality of the fishes. Suppose in certain environments, we are coming across a high concentration of fish, then it can be taken as an indicator of good water quality. In addition to the entire trophic levels, we can also make use of the process rates as indicators. What I mean by process rates is the primary production determinations. How much carbon is produced, gram carbon per meter square area or meter cube area per day or per year, we can take into consideration. That way, we will be able to monitor the environment. And there are also other organisms which can be used as indicators of the marine environment based on their ecological strategies. Here I have given two indices. The first one is the nematode copepod. In nature, many organisms are opportunistic organisms. Many are conservative organisms. In the absence of impairment, the conservative organisms will be thriving well. When impairment is starting, slowly they will withdraw, they will disappear, giving place to the opportunistic organisms. So that way, copepods are conventional organisms. And many species of nematodes are there, and they will be able to thrive well when there is impairment. So that the index is being used as the indicator in environmental monitoring. And again, these two groups of organisms, that is polychaetes and amphipods. Amphipods are conservative organisms. And when impairment is starting, immediately they will disappear. And with the return of the pristine conditions, they will appear again. So that way, amphipods serve as very good indicators. Polychaetes, there are many opportunistic species, and based on their presence, we will be able to understand the quality of the environment, health of the environment. So this is the basis, ecological basis. Under normal situation, we are able to see quite a large number of larger organisms also. When impairment is starting, slowly they are withdrawing. We come across this transitory stage when we are not able to see the larger organisms. And then ecotone, that is transition zone, is going through the ecotone. So drastic changes are taking place. And on the left-hand side, we are able to see the peak of opportunistic species. Only small organisms are there. And with the impairment increasing, there will not be any organism at all. So based on this simple ecological observation, ASDI, Tecnalia, an institute in Spain, many of our friends will be remembering Professor Vijay Nandan. He invited a scientist, a very famous scientist from this institute. He founded this index, AMBI, based on that ecological observation principle only. He divided the invertebrates into 5 groups as highly sensitive, indifferent organisms, then tolerant organisms, second order opportunistic species, and first order opportunistic species. And color convention is being used. If in a particular place green and blue colors are dominating, then the environment is healthy. There is no any impairment. But on the other hand, these colors, red and other things, if they are dominating, then there is impairment. So this way, making use of the biological indicators, we will be able to undertake environmental monitoring. And how we will be able to monitor the physico-chemical parameters and also contaminants. For monitoring the contaminants, very good methods are there. Instrumentation facility is also very good now. So now let us see, we are all academics, we started in a very simple way. And suppose I am doing work in the industrial area, how I will be able to find out the temperature of the water, surface water? I will be making use of a thermometer. We go to a hospital when we are having fever. Once upon a time, the doctor, not the doctor, the nurse will be making use of a thermometer. She will be pushing that one into her mouth, asking us to hold for some time, and after some time she will be taking out and looking at the temperature. She will be noting down and that will be sent to the doctor. So just like that, we immerse the temperature into the water, then we note down the temperature of the water. Then sediment also, we insert the thermometer into the sediment, then we note down the temperature of the sediment, right? In the industrial area, it is quite all right. Suppose I am lifting a sample from a depth of 15 meters or 30 meters. It has to come the whole way. I am making use of a grab. So as soon as the grab is coming up, I try to insert the thermometer into the sediment. It has lifted from the depth. Will the temperature be the same? Will the temperature at the bottom of 30 meters and the temperature once the sample is reaching the surface will be the same? Naturally, some difference will be there. Same way, I monitor the salinity with the help of the refractometer. And many scientists, they do not accept the value of refractometer. But when we have to analyze quite a large number of samples, and when we are calibrating this instrument periodically, it has to be accepted. In the same way as sediment, I am trying to find out the salinity of the water at the 30 meters depth. So the water is also coming along with the graph. And the water is, once it is lifted, some water will be falling down. So I am taking that water, I am taking a drop, I am putting it here. I try to read the salinity. Will it be the same? So that way, drawbacks are there in the conventional methods of monitoring. Again, pH pen. In the industrial area, it is quite all right. But recording the pH of the sediment taken from the depth of 30 meters, it may not be that accurate. Transparency, of course, we'll be able to measure exactly well with the help of the CGDs. Same way, turbidity meter. We collect water, and there are difficulties in collecting water from deeper depths. But anyhow, when we are collecting, we have to measure individually. Sample by sample, we have to measure. Then the sedimentation rate also, making use of this frame, we will be able to measure. So all these things are possible in the shallow waters, in the inshore waters, but in the deeper waters, things are very difficult. So these are all the constraints we are able to see in traditional monitoring. Oxygen and salinity analysis. These are titration based. So if you are collecting quite a large number of samples, we have to spend quite a lot of time at the titration. And moreover, the researchers cannot cover a large area. Suppose I am studying a particular organism in Vembanar Lake, I can fix the maximum 5 stations or 6 stations. I can cover every month. Every week it is not possible. Every fortnight it is not possible. It will be cumbersome. If I have to analyze all the samples, make use of the traditional methods. So what is the way out? So now the monitoring has been augmented with an array of innovative, new and technical approaches. Instead of titration, if you are able to know instantly the values of salinity, temperature and other things, it will be quite ideal. We will be able to cover a larger area. Large number of places can be covered in no time. So these are all the advantages. We have to make use of the technological advancements. So now the objective of the marine monitoring has changed. So what is the objective? It is to strike a balance between growth. By growth, I mean economic sustainability and also conservation. And what is the process I have to make use of in the marine monitoring? I have to make use of the process that is ecosystem approach as enshrined in the Convention on Biological Diversity. I will be able to make use of. It will be much more effective than the traditional methods. And what is the ultimate aim of marine monitoring? To have a clean, safe, healthy, biologically diverse and productive seas and oceans. So let us see in the place of conventional methods what we can do. So this NEAT has come into being. The expansion of NEAT is Nested Environmental Status Assessment Tool. So from where it has come, it is quite interesting. It has come from the laboratory of a scientist whom was invited by our respected professor, Dr. Vijay Nandan. It has come from the laboratory of Dr. Borja. They have perfected that one. So amphiphod polychaete ratio, it is a single item. Nematode copepod, it is a single ratio. It will not give an integrated picture. To get an integrated picture, to get a comprehensive picture about the impact of a particular habitat, they have devised that one. So this is the opening page of the NEAT, the open page. It is a nested environmental status assessment tool. Here what they do, they make use of biodiversity assessment over the spatial assessment units. They cover the habitats, they cover the ecosystems, they make use of the indicators to assess the quality. They will be able to get an integrated value, not values for bendas, for values for fishes. Individually, they will not be getting. They will be able to get an integrated value. So how it is possible? So these are all the habitats they cover. They cover the pelagic, they cover the benthic, both they cover, and they take into consideration various components. They take into consideration the fishes, the birds, the mammals, the pelagic organisms, benthic organisms, even primary productivity they take into consideration. And this is the scaling. They have 5 categories as in AMBI, that is high, good, moderate, poor, and bad. So this way we will be able to undertake environmental monitoring the modern way. And this is the final results. So individual results are given. Please look at here. This is the value for the fish, component fish. These are all the values for the component birds. These are all the values for the component mammals, pelagic organisms like this benthic primary production and all. And integrated values are given here. So these are all integrated as a neat value. And what is the status also, it is indicated with the help of the color combination. So green, it is good. And high also we are coming across at the bottom. And there are moderate situations. So what we are able to infer is an archipelago, that is little archipelago, is having a good environmental status. So this is good. We have to make use of this one rather than the old and the conventional things. And coming to monitor the environmental parameters, we can make use of this OSI multi-parameter sonde. The water samples for titration. Here we can see a lady scientist is sitting in the intertidal area. She is letting down the sensor into the waters. She is looking at the values in the display unit. And if there are 2 researchers, they can do the work combinedly. The coral reef area also, or in some other environment, you can dive, you can lower the instrument, you will be able to record all the values in the display unit. Profiling also, you will be able to do very nicely. Why we must waste so much of time in titration and other things? Of course, we have to do the calibration before setting out for sampling and all. So these are all the sensors. This is the display unit. And what are all the parameters we will be able to cover? We can cover conductivity. Of course, through conductivity, we will be able to get information regarding the salinity, temperature. These are all the things conventionally we have been covering. And whatever may be, whether modern monitoring or conventional monitoring, these parameters matter. Depth, dissolved oxygen, dissolved organic matter, pH, in the changed circumstances of ocean acidification, it is becoming important. Turbidity in coral reef areas and all this is becoming important. In addition, we can get information regarding the optional parameters like rhodamine, chlorophyll, phycocyanin, phycoerythrin and all, and also these ammonia, ammonium, pressure, blue-green algae, chlorate, nitrate, total dissolved solids, total suspended solids. And the units will be able to save the data. We not note down in the field itself, we can come back, we can retrieve the data, and we can do the analysis. So this is good, a little expensive, but a progressive head of the department like Professor Vijay Nandan, with the help of distinguished dean and all, they will be able to get this instrument for the department. And also the habitat mapping. Suppose I am carrying out a study on bendas of Villarishiri, I never used to bother about the habitat mapping and all. But it also gives very good information. What I have to do is I have to lower this unit That way I will be able to know drop-down cameras I have to make use of. I can drop it down. I can take the pictures. That way I will be able to know what type of habitat is there in the bottom of the Velar estuary. We have not been doing this one, but time is changing. We have to do this one also in the days to come. And the turbidity loggers are there. So a coral reef area is there. We have to continuously log the turbidity. So for that, this instrument we can make use of. It is good for unattended turbidity logging. And then sound, we have to record the sound also. So times are changing. In the ocean, the sound is increasing. Natural sound is there. Fishes and other organisms, whales, porpoises, dolphins, all are making the noises. But we have added much more sound through piling work, by ship movement, by carrying out various construction activities. We have increased the sound. So we have to monitor the sound also. That is mattering now to a greater extent. I remember Professor Menon, whom in association with Professor Damodaran, they organized a conference on Muscle Watch. Muscle Watch was a very famous program. The whole of India was very much interested in the Muscle Watch. We used to collect mussels from the field. We used to study the bioconservation. In the laboratories, we used to expose them. And that way, we studied the bioaccumulation of various metals and all. The drawback of making use of live material is there will be changes in relation to the size groups. There will be changes in relation to the maturity stages and all. So how to overcome that deficiency? A scientist thought. And that way they arrived at the artificial clamp. They call it as a chem catcher. So this can be made use of. It can be used in the field. It can be used in the laboratory for exposure studies and all. So this is the way the analysis is being done. The analysis remains the one and the same only, just like we analyze the clamps, muscles. We are going to analyze this one. So this is retrieved from the field. Then the extraction of the contaminants is done. Then the measurement is done with the help of the ICP-MS. MS. So this way, in the place of live organism, we can make use of the artificial clamp in studies. And now, having covered the implements we can make use of in the intertidal and the inshore areas, now we are moving to the offshore area, and how we'll be able to do effectively offshore monitoring. We can make use of the platforms. We can also beam sonar array and all. That is, platforms can be divided into two types as moving platforms and fixed platforms. So first we will see what is the moving platforms. And these are all the examples for moving platforms. Our Sagar Sampada is a moving platform. And the other voluntary observing ships. And throughout the world, ship movement is quite heavy. They carry large cargo. And vessels can be made use of marine monitoring. These are called as voluntary observing ships. What they require instruments. We can give them, they will fit in the vessels. The instruments will be collected. So the ships, what they require, They require the instrument and also they require the forecast. In their route, if any storm is coming, they have to be informed well in advance. So that is a very good partnership. So these vessels are used in large numbers. And also ships of opportunity, ordinary cargo, small vessels can also be, depending upon our requirement, we can make use of ordinary cargo ships also. We will be able to have repeated measurements. Suppose you are going for a cruise on Sagar Sambhada, only through a particular route we can go, we can collect the data, we can collect it throughout the world. So we will be able. Moreover, it is cost effective. We don't have to operate the vessels. The vessels are carrying cargo, so they are not asking any money. Only the warning and all they are expecting from us. And a long-term continuous plankton recorder also we can make use of in such moving platforms. And the data on marine air temperature and ocean acidification can be collected quite effectively, making use of the moving platforms. This is the way we deployed the CTD in Sagar Sambhada. Our leader is very much here. She knows very well. With the help of the Sagar Sambhada, Dr. Professor Vijay is also a part of all these exercises and all. Everybody knows that CTD is being operated in Sagar Sambhada. We'll be able to get information regarding the conductivity, depth, and also temperature, and other parameters, also water samples and all, we'll be able to get with the help of the CTD. One problem with the CTD is, it will not be touching the bottom at all. It will be remaining above the bottom. But anyhow, that temperature will be recording as the bottom temperature or sediment temperature. Once a sediment sample has been lifted from 1000 feet, if you are inserting the thermometer, that will be very unfortunate. So this reading we have to make use of. So ordinarily a ship will be having all these implements. It will be having the multi-beam echo sounder, and we will be able to get 3D maps of the seafloor. And we can make use of in all our studies. All these days we never bothered about these things. And thanks to Professor Rosama, we used to keep the maps of the echo sounder in our thesis and other publications when the referees permit. And the ADCP is there. It will be able to profile the current and all. And the topography, ordinary echo sounder, it will be able to give information regarding the seafloor topography. And the CTD is also there. To this, what we have to do is we can attach a ferry box, and many vessels will happily carry this ferry box. And what is the advantage of this ferry box? We will be able to get information regarding salinity, pH, oxygen, turbidity, then chlorophyll, besides all the nutrients. That is nitrate, phosphate, silicate, and also ammonia. So this way, throughout the world, from the entire ocean, wherever ships are moving, wherever the ships are taking the cargo, we will be able to collect the data. So very good arrangements are in place throughout the world. And the continuous plankton record. One scholar in our center, he completed his PhD degree. He concentrated on algae. Then he moved to United States of America for another PhD. When he came in between, we were discussing. So here he worked on algae. There, what problem you have chosen? And he was telling, here I was working on algae of LR estuary, a small estuary. There I am covering the entire Gulf of Mexico. Then we were taken by surprise. We were asking how you will be able to collect samples from a large area. Then he was telling, we are making use of the moving platforms. We give them the continuous plankton recorder. So the ships, the vessels will be collecting the plankton continuously. And they will be putting formalin. It will be preserved. It will be towed at a depth of 10 meters. Then every 10 nautical kilometers it will be taken out. The plankton will be retrieved. Again, it will be deployed. That way, continuously, plankton can be recorded. So this way only, I am collecting the plankton over a large area. So these are all remarkable advancements. We can also make use of, instead of collecting samples in various places, 5 or 6 places, and analyzing. Only funding has to forthcome, but people are in right places like Professor Vijay, Professor Rosama, and for helping Professor Sarama, Professor Hatha. They are all there. They will be able to mobilize funds for getting all these things. These things are quite possible in the days to come. Then ROV, NIOT operated the ROV, but these remotely operated vehicle was used only opportunistically and not routinely. And anyhow, this is good for making measurements and observations in places which are very difficult to dive and collect. And these are all attached to a ship or to a vessel. And these are all operated by a person, by an active person. And the whole thing will be under his control only. The cable will be supplying power and the data collected will be transferred to the main unit. And that is the only advantage. That is, in very difficult environments, we'll be able to collect the data without ourselves diving into the area. This is the way the ROV will be looking. In Chennai, in NIOT, we saw this unit, but I do not know how far it is being used now. And then we have the autonomous underwater vehicle. This is better than the earlier one, ROV. However, it is sharing most of the capabilities of ROV, but it is under its own propulsion. That is the advantage. And the range covered is quite extensive. It will be able to cover a large area. It can cover thousands of kilometers. Even it can descend to the depth of 100 meters. That is the advantage. And this is found to be very good for ecosystem monitoring. Then we have the autonomous underwater. This is the way the autonomous underwater vehicle will be looking. And in its place, we can also make use of the gliders. And the gliders will be gliding. They will not be drifting with the current. So they will be moving at the subsurface waters. And they will be operating independently of the ships. But they will be communicating via satellite, enabling data upload and also mission planning and updating. So gliders are also there. So this is the way the ocean robot glider will be looking. These are all quite expensive, but still as an institute, they can organize and we will be able to participate. We can get the data and they can collect the data and we will be able to share the data. And alternate to glider is also there. That is the unmanned surface vehicle. Here the wave power is used for propulsion. There are also internal solar batteries, and these are all used for navigation, communication, and also for attending to sensor payload. And make use of this one, we will be able to collect information regarding the water quality and passive and also active acoustics, and we can do also current profiling and all. And this is the way the unmanned surface vehicle will be looking. And I do not know whether any institute is making use of this vehicle in India. Then we have the remotely piloted aircraft. It is very good. We can make use of, just like the drones. It is a small flying vehicle capable of carrying various sensors. Typically, some form of camera system is there. So it is equipped with both hardware and software for navigation, control, and data acquisition. It is very good for vegetation mapping. Vegetation dynamics will be able to do, and industrial area will be able to cover. Shallow water areas also will be able to cover very nicely. Just like drones that we have been making use of in marriage halls and also in other places for regulating traffic and all. This is the implement, this is the way it will be looking. It may not be that expensive. The whole of Cochin waters can be covered. The whole of Vembanad lake area can be covered nicely. And as a project, it can be large and it will be very good. Now I am moving to the other one, that is the Earth observation. Earth observation, so many satellites are orbiting. And with the help of the Earth observation systems, we will be able to collect data regarding the chlorophyll A, suspended particulate matter, turbidity, and also other dissolved organic matter and all. We will be able to collect. As I mentioned, so many satellites are orbiting the Earth. Depending upon our requirement, we will be able to input the parameter. We can input the required sensors. That way, we will be able to collect the data. And these are all the sensors used for collecting information regarding various parameters: salinity, temperature, oxygen, turbidity, chlorophyll, fluorescence, light, and the nutrients. So, very good information we will be able to collect, make use of the sensors. Suppose we have a requirement for a particular place, we may not be, the satellite may not be orbiting that area. That problem is there. But there we can make use of other methods and collect the information. And coming to the future possibilities of Earth observation system and the various functional types of harmful algal bloom, this is our requirement. So can be found out, and based on the color also, we will be able to identify the dissolved organic matter also. So these possibilities are there with respect to the Earth system. Now coming to the fixed, moving we have covered. Now we are coming to the fixed, and many good things are here also. So what are all the fixed point marine observation system? We have the fixed moorings, buoys in Cochin backwaters, during the channels, and all buoys are there. There we can fix the sensors. We will be able to collect the data. Probably many are collecting the data, and fixed piles can be made use of, towers, seabed landers, all these things can be covered. The advantage of this one is, suppose a ship is moving and in that place an episodic event is happening, we may not be able to record. So, but anyhow, when we are making use of fixed point marine observation system, we will be able to record such things also. So events that are having high temporal variability also, we will be able to cover. These are all the buoys, the fixed piles, and these are all the towers we can make use of. These things will be expensive, but with the help of various other institutes, we will be able to do it. We can deploy so many buoys along the Cochin backwaters, and the data can be shared by various institutes. And Cochin University of Science and Technology will be able to make use of such data to a greater extent. The scientists in other institutes may not be having time to make use of the data. So we can make use of the data to a greater extent. What is required is to strike some agreement. So these are all the parameters. All the parameters we require, we can add the sensors, that way we'll be able to collect the information. And coming to the practical application of the data collected, I can cite one example. Such data are used for strategic operations. And one example I am citing is the UK Coastal Flood Forecasting Service. It has been doing it very nicely. And the other advantage of such data is for designing the coastal sea defenses and also the offshore structures. Based on the data, we will be able to design all these things. And Argo float you might have heard about, and it has revolutionized monitoring. It started way back in the year 1999, and now we have a coverage of 5,000 floats throughout the world. They collect information. The upper 2,000 meters, it has been covered very nicely. Nowadays, we are getting alert. Next to 2 days, it will be hotter. Next to 2 days, it will be chiller. Like that, we are getting information. Thanks to the Argo float, we are able to give such forecasts. So it is a very good arrangement the world nations have done. So see the deployment of Argo floats in the whole of the oceans And this is the way it is working. Every 10 days, it will be surfacing. It will be transferring the data to the satellites, to the land station and all. And India's tsunami early warning system is also based on fixed platforms and all. Here we have the seabed monitor. It is collecting the information. It is passed to the moved by, and in turn it is passed to the satellite, and that is from the satellite it is passed on to the land stations. So this way it has been working quite effectively. And high frequency radars can also be made use of. In Cochin area, one radar can be made use of, either in QSAT or in other institutes. It can be fixed. It is a fixed unit. It will be able to cover a very vast area, 100 kilometers it will be able to cover. And whatever parameter we require from the sea, we can collect. And that is the advantage of the high frequency radar. The unit is very simple. This is the unit, very simple box and a structure like this. And even this is looking different. See this box. So many institutes, the instrumentation is coming from United States of America. And China is also making a CODAR. Out of curiosity, one institute which obtained CODAR from United States of America, it opened it to see how it works. Out of curiosity, they opened. Then this act was brought to the attention of US company. What they told, hereafter we will not be cooperating with you. Their fear, it is a very simple instrument. If you are able to study how it works, you will be able to fabricate. But the original company is not for that one, so they have entered the contract with that institute. Like that, so many innovations we can also do. This way we'll be able to fabricate the needed requirements. Our requirements may be different from theirs. So coming to the tail part of my presentation, we have to strengthen the taxonomy. Taxonomy, many feel that it is an obsolete subject. It is not a very famous subject. So if you are working on taxonomy, you have to spoil the health and all. That thinking is also there. But there is a very good need. And why I am telling in particular is, many MSc students are attending this one. That's why I am making it as a point. Nematode taxonomy, our Professor Vijay is concentrating on nematodes. And if youngsters are able to assist him, we are able to prepare monographs on Indian nematodes, keys for identification, keys for easy identification of the nematodes. That will be a very good contribution to science. Same way, benthic copepods, there is no any good manual. There is no very good manual for nematode also, Indian nematodes I mean. Same way, amphipods, there is no very good Indian manual for the identification. We do for a particular place, it may not be holding good for the whole of India. Polychaetes, still we make use of a day which was published in the 60s. And the invertebrates at large, we are not having a very good key for identification. So youngsters, we can concentrate on the group of your choice. You can strengthen the taxonomy of these groups. And you cannot become, by this act, you may not be becoming an expert in a dry subject. You will be able to complement with this one. In addition to working in the museums, in addition to examining the herbarium, in addition to examining the specimens under the microscope, you can also work in the wet laboratories, extract DNA, amplify, then barcode, study the genetic diversity and all. Then in addition to the wet lab, you can also sit in the dry lab and you will be able to map the whole things. So based on the external characters, how will you be able to identify? You are barcoding it. With the help of the barcoder, how you will be able to confirm the identity and all. So that way you can become both an expert in the old field and also in the new field. But I should not tell that that is an old field. Even today, species are being described, not only from land, from the outer space also, species are described. So youngsters, you have the time, you have the energy, you have the drive, you can do that one. And most of the technological innovations, they are coming from the other countries. I mentioned about Kodak. And OSI, it is also from some other country. Artificial clam, long back we started a program on muscle watch and all. And we could have found out this one. The buoys, floats, any instrument we have to import, ADCP, ROV, AUV, ferry box and all. These are all the inventions of other scientists. And we have the technical manpower. And the Microsoft, it is being headed by an Indian. And what we lack, we have everything. We have the expertise. And if the senior members are facilitating, if the leadership is facilitating, political leaders also facilitating, and good technological innovations will be coming from our country also. It is our hope, and it will be a very good tribute to Professor Menon also, who served the cause of environment, who did quite a lot of work on pollution and environmental monitoring. And even this model, this is oil spill model. In Kochi, we have the refinery. Suppose oil spill is happening, and how it will be affecting the areas, how much area will be affected, up to what extent the beaches will be affected and all. These are all done with the help of the models. But again, for doing these models, we have to go overseas. We have our own models, but all these things came from the other countries only. So we have to design our own models. We have very good mathematicians. We have very good software personnel and all. So we have to do quite a lot. Youngsters, I appeal to you, please take interest. Definitely you'll be taking interest and you'll be able to contribute technically. So what we have to remember, thanks to scientific advancement, the monitoring methods for the marine environment will be very different 10 years from now. 10 years back, 20 years back, we started with the thermometer and titration for salinity, oxygen and other things. Now where we are standing? Due to scientific advancement, so many instruments are there, and we will be able to collect effectively larger area and all. 10 years from now, scientists are telling that things will be quite different. If Indian scientists are able to contribute much more, that will be a very good tribute to Professor Menon. So with this, I conclude my presentation. I am thanking again Professor Vijay for the wonderful opportunity. I am thanking the distinguished Dean, Rosama Madam, and also our other teacher, Professor Sarama Madam, and all the other learned faculty members of Department of Marine Biology, Microbiology, and Biochemistry of Cochin University of Science and Technology. Professor?
Hi, sir. Yes, sir. It was a very informative.
Thank all of you for your attention.
Okay. It was a very informative, exhaustive presentation. It took more than 1.5 hours, I think. Okay.
Thank you, madam. Thank you.
I've taken a lot of pains in the preparation of the slides.
Just started, madam. Thank you. For your leadership, for QSAT, anything can be done, madam. Thanks to you. Thank you.
Okay, thank you, sir. So now it's a time for discussion. Now the floor is open for discussion. You can give your questions in the chat box also. Sir, this NEET software, is that free or we have to pay for it? NEET.
We have to pay for that one, madam.
Okay.
And the same institute, the ASDI Technalia, it has perfected that one. We have to pay. As an institute, we can pay, madam.
Okay. How much? Any idea, sir? How much will it be?
At present, I do not have, madam. Anyhow, I will be able to get in touch with Dr. Jai. Dr. Jai Das, our good friend, and I will be able to get the information.
Have you used it, sir? Have you used it?
I saw it. I heard the presentation. I saw the presentation, and I felt that it is very good. It is integrating various indices, various taxonomical units, various groups of organisms, various habitats. It is able to integrate. That way, in a single value, we are able to know the quality of the whole ecosystem. That is the advantage.
Okay, can we use the partial data also for the analysis, or is it mandatory that all the different aspects or inputs are to be given for running of the program?
Whatever data we have, we can make use of it, madam.
Okay, right, sir. Right.
There is a question from Dr. Surjit, madam.
Oh,
Surjit.
right. Okay.
Dr. Surjit is raised his hand.
Okay.
Oh, sorry. I have not that much audacity to ask my sir the question, but I would really appreciate this time what is being asked, delivering this wonderful talk, which actually reminds me our my PhD days when I used to in Sagar Sampada and data from during these benthic studies in association with Sama Madam and Sarama Madam and Damdaran sir. So thank you so much, sir, for motivated.
Thank you.
From the young
Thank you.
The taxonomy field, which is really frustrating. People, even more scholars, people give them the work on that not comfortable.
Yes, that's true. Yes.
But your talk has motivated, and definitely, I think various new people will use taxonomy because
Thank you.
we cannot progress further to identify what kind of pollution are happening even in our system, and we cannot actually monitor.
Yes,
We have learned new techniques like NEAT and other things which we can make use for monitoring these marine pollution. Thank you so much, sir, for your wonderful and informative.
Thank you, Dr. Sujit. Thank you.
Anybody, any other questions?
Some audio is coming now.
Yes, madam.
From where it is coming?
Audio. Some audio is.
Oh, some sound. You mean?
Yes. Some.
Yeah, from where it is?
I don't know, some external audio.
I am saying. Yes, madam. This is the call for prayer. Being Friday, They are giving the call in advance.
Oh,
Probably
okay.
that
That's
may be
why.
the.
Oh,
Oh, sorry,
Okay.
sorry. Now
Nice.
it has ended. Yes.
Yeah,
Yes.
nice,
Okay.
sir. Thank you.
Okay. Okay, any other questions from the audience? Sarama teacher? Sarama. Vijay sir? No.
Yeah, sir, thank you so much. Teacher also, thank you so much. And sir, it was a
Thank
revelation
you.
for me.
Thank you, sir.
Yes, sir, it was a revelation for me, sir, going through the and hearing
No,
your
no, no,
very
no.
because hearing your esteemed and very thoughtful lecture, because it is, we should not undermine it, sir, because there are many things which we know, but there are many things we are still yet to understand, because especially the tail spark which you have left is very pertinent. Very pertinent, sir,
Okay.
because we don't have taxonomy.
Okay.
Then, of course, the what Borja has developed. Borja, in fact, we could meet him when he came to India last time for our meeting. And
Yes, sir.
what
Yes, sir.
Borja
Thanks
has
to you.
developed for
Thanks
the
to you.
yeah,
Yes.
yeah, thank you, sir. What Borja has developed for monitoring of marine environment, especially using animal models, marine organism as models, was really exemplary. And he has now come up with
Yes,
a whole total package,
yes,
and it is very useful. You
yes.
can simply download it from the website, and it is the latest version. As you suggested, many things have been put in a nutshell for the youngsters and the old. That is the hallmark of this lecture.
Yes, sir.
Yes, and so much painstakingly,
Yes,
sir, has given it. Kudos to you, sir, and with a great pronouns to you on this great memorable day when Professor Menon. He was a enlightenment
Yes.
for all of us. Enlightenment, a thorough
Yes, sir. Yes.
professional in his approaches, and
Yes,
great
sir.
researcher in the marine environment and marine uh, monitoring,
Correct.
especially in the marine monitoring,
Yes, sir.
especially ecotoxicology, marine toxicology.
Yes,
He has left his footprint not only in different areas of marine
Yes,
biology.
sir.
So it is a tribute that
Correct, sir.
especially a doyen like you could address this webinar, and we are all fortunate, sir, honored for your presence. But I just want to ask with this
Thank
small
you, sir.
background.
I also feel honored, sir.
Yes, sir. Thank you, sir.
Right, sir.
So
Thank you,
very
sir.
fortunate that you have.
Thank you, sir. Thank
It
you.
is just accidental, because I never thought that it was the third anniversary of his memorial anniversary, because it was just a coincidence when our dear teacher, Rosamond Madam,
Yes, sir.
informed me today
Yes,
morning. It was
correctly
just,
pointed.
I would say, it's providential. It's
Right.
providential
Yes, sir.
that we have the good blessings of Professor Menon for the
Yes, sir.
department and for the
Definitely,
good
sir.
good work we all are embracing in the field of marine biology. So that with
Yes,
just
sir. Yes, sir.
one point, sir, I was just asking. Sir, why don't we India
Yes,
have
sir.
a
Yes, sir.
policy, sir, for monitoring? We don't have a specific policy. Government and national agencies should,
Yes, sir.
because even I will say that let us have the Central
Yes,
Pollution Control Board, pollution PCBs. Then we have the Open
Yes, sir.
Marine Act we have. Then the Wetland Acts we have. So many
Yes, sir.
acts and rules are there.
Yes.
But we don't have a policy
Yes,
for marine monitoring. So why not this
yes, sir. Definitely.
this points are very important, that we develop a policy for the country, marine monitoring policy,
Yes, sir.
and what are the guidelines, what are the
Policy. Yes.
stipulations. This is very important, sir, and it will be,
Right.
if it comes up as a policy decision, a specific policy decision, sir.
Yes, sir.
The criteria based, I don't know,
Right.
sir. Different criteria are available in India. We have borrowed it from
Right.
EPA, EP, and so many other sources. And if it's possible,
Yes,
if we are able to
yes.
make a policy for the country, sir, with people like you, sir, as a joint, it will be a great. Is there anything, sir, your knowledge on that? Could you just share some?
The earlier one initiative, that is COMAPS, was there,
Yes.
but unfortunately it has been disbanded, and no other project is moving towards that direction. So
Yes, sir.
as you pointed out, it will be a very good thing, sir. Yes.
Policy, sir. I'm been not just monitoring, sir. A policy for the
Yes.
country. Because-
Yes. Policy for the country is definitely essential. And following that, if we are initiating some programs also, that will also be very nice. Yes.
Yes. Thank you, sir. Thank you so much. And,
Thank you. Thank
very
you.
great.
Thank you. Thank you
Great,
so much.
sir.
Thank you.
Sir,
Thank you.
teacher,
Okay,
thank you,
there
teacher.
are,
There's a question.
Thank
there are
you.
more, questions. Let
Yes,
us
there is a
find them,
question.
sir.
I think Abhijit is asking a question, teacher,
Okay,
in the
okay.
chat box. Chat box, I think.
Chat box. Abhijit, yeah, sir. It was a very good presentation. I have a question. Do you think that dilution of environmental impact assessment would have an adverse effect on our oceans when some development
Definitely.
project is coming up? Okay.
Definitely. Definitely. Definitely.
Okay, sir. Hope there are no more questions. Yeah. Sir, it was a wonderful presentation. You have given an overall view of the anthropogenic activities leading to the alteration and degradation of the marine ecosystem, and the necessity of monitoring the environment for sustenance of the marine ecosystem
Yes.
with its all biodiversity and beauty. And the various tools, physical, chemical, biological, to be employed in monitoring of the health of the ecosystem. And from the simplest tools like the pH meter, turbidity measurement equipment, and the most advanced ones like ROVs, AUVs, USVs, and all those things you have discussed, radar systems. And sir, thank you so much for spending your valuable time in delivering this lecture, and especially as Professor Bijoy said, the tail spark, that's a term you used, I think, some tail spark. The strengthening of the taxonomy, a subject which is not sought after by many, but I think it is getting momentum. And now there are a lot of youngsters coming into the taxonomy field. And with the advent of molecular taxonomy, it has become more attractive nowadays. So they can be experts of both the conventional as well as the molecular biology or molecular barcoding. And thank you so much for your advice, sir. And thank you so much for spending your valuable time in delivering this lecture. And I would like to thank Professor Bijoy for organizing this lecture in honor of late our beloved Professor N.R. Menon on his third death anniversary. May I also thank Dr. Sarama, Dr. Surjit, and all those scientists who are online, all our faculty members, scholars, students, and all other participants. And thank you so much. And at the last, let us pay our respectful homage to the late Professor N.R. Menon, who has been a guiding force to the marine science community for over 4 to 5 decades. And thank you so much. Thank you, everybody. Thanks a lot once again.
So thank you, sir, for the very informative presentation, Ajmal sir.
Thank you.
Thank you.
Thank you. Thank you. Thank you. Thank you.
Thank you.
Thank
you,
you.
Bijoy, sir.
Thank you.
Rosama teacher.
Thank you. Thank you.
Okay,
Great.
bye.
Bye.
There was a very good audience.
Yeah.
Around 88 people were there.
Yes.
88,
Yes. Yes.
yeah,
Yes.
participants.
Yes.